Hello, I'd like to thank our speakers. My name is Claire Orkim-Hasty. I'm Scientific Director at Scientific Education Support, and I'd like to welcome you now to put forward some questions to our panel. And if you'd like to do that through the question and answer button on your screen, I will be facilitating reading those out to our panel. On the line today, we have Dr. Jesus Bañales, we have Professor John Bridgewater, Professor Shahid Khan, Mr. Andrew Murphy, and Helen Morment. So if you'd like to send in your questions now, we can start the panel discussion. So I'd like to start with a question for Jesus here around how better CCA coding could actually affect ongoing research. How could it benefit ongoing research, Jesus?
Thank you, Claude. First of all, thank you for the invitation and congratulations for this very important initiative. It's definitely very welcome for the cholangiocarcinoma community. In terms of research, I really truly think that this new classification will be very helpful for both experimental and clinical studies. For instance, this will allow us to characterize better the tumor biology of the different subtypes. We will be able to understand, or at least in part, the similarities and differences between each cholangiocarcinoma subtype that before was not obviously done. We have available different human cholangiocarcinoma cell lines, so it is important to understand if the molecular mechanisms altered are general or specific to each subtype, and also obviously considering the genomic and genetic profile. We have available different animal models of cholangiocarcinoma. It is important to compare the different animal models with the different human subtypes and try to define and see which is the best model that mimic each subtype, because this might have important translational impact. So we can see if the responses to the new therapies in the different models are general or specific to each subtype. And this definitely might have important translational impact in the design of clinical trials and in potential sub-analysis in clinical trials. Regarding clinical studies, it's very important also this new coding for the registry databases. So in the European NSCAR registry database, we have adopted this new coding. So it is important to understand the similarities and differences on risk factors between these three subtypes, their mutational profile, their incidence, their management, as well as the response to therapies and prognosis. So this is some studies that we are currently ongoing and trying to see these potential differences and similarities. And again, also, well, when we evaluated and design and evaluated clinical trials, potential sub-analysis can be conducted, evaluating the response to therapies, considering all three subtypes. And I think this is a very important point, because the biology and the risk factors associated to each subtype is in general different, or might be different, and the potential response to therapies also could be potentially different in certain cases.
Great, thank you, Jesus. And John, coming to you, given the importance conveyed there by Jesus, how do you think we can encourage more clinicians to put patients forward for molecular profiling?
Thank you. Well, one of the targets that we find in primarily intrahepatic cholangiocarcinoma is the FGFR gene, and this is altered in about 1 in 10 cholangiocarcinomas. We have very good drugs for that, and indeed one of them, pemigatinib, has recently been NICE approved. As such, it's now effectively mandatory for every patient with a cholangiocarcinoma to have the FGFR fusion looked for. There's a bit of detail here that's quite important. The FGFR fusion is looked at using a test called RNA-seq primarily. The FGFR, the other abnormalities within that allow targeted therapies within cholangiocarcinoma, they tend to use a different technology. And so I think it will be a little while before everybody has all the testing that they're required. But it is a question of education, primarily of the doctors and pathologists looking after patients, and just the knowledge of this potentially extremely good treatment working its way through the system. Nice guidance always takes some time to settle in, possibly sometimes years to settle in. But for patients, the sooner we can do this, the better. So in short, it is a question of education for everybody: patients, doctors, everybody out there. And AMMF has a key role in really waving the flag here and raising awareness of this very important innovation.
Thanks, John. Brilliant. And so you mentioned AMMF there, and over to Helen. How do we think that better coding could actually improve the outcomes of CCA patients in the EU?
Well, I think it's supremely important, as has been very succinctly and well explained, really enlightening presentations this morning. Thank you so much to everybody who presented. Everything that we've heard this morning points towards the difficulties that we've had and we still have because of a lack of robust data. And this all comes down to the coding problems and the fact that it's so difficult to prove what we at AMMF and many other people actually see. And I think it was Hassan who mentioned, no, I beg your pardon, it was Shahid who mentioned something like 1%. We're seeing 1% of perihylers actually recorded, when in fact most people will tell you it's well over 50%. And I think Hassan said even 60% of cholangios are perihylar. So what we're having at the moment, what we see at the moment, is a disease with an increasing incidence, one of the worst prognoses out there. And that's something that hasn't changed in decades. We need to improve the situation. We now have targeted therapies. And I had to smile quite wryly when John said, quite rightly, it is mandatory for every patient to be examined, to have molecular profiling for the FGR fusion. That very much depends on the doctors diagnosing. Will they put the patient forward? Do they know about it? And this all comes down to this lack of awareness of cholangiocarcinoma, because data show that it is, the incidence is very low. So there's really very little interest. Actually, the incidence is not low. So if we can get better coding, which is coming with Icd-11, therefore better recording, we will then have better data. And with that better data, we have the ammunition in Europe, in the UK, in fact, globally, to say, look at the incidence of this cancer, look at the mortality, which actually parallels incidence, and let's really work together to raise awareness, to raise interest, to boost research and improve the future for all those unfortunate enough to be diagnosed with this disease.
Thanks, Helen. Thank you. And going over to Shahid there, so we heard there about the importance of documenting those molecular factors. But what's the importance in terms of the epidemiology of those different factors influencing CCA and the different subtypes?
I think the importance, it's important to track diseases. One of the reasons is the importance of diseases given when it comes to resources, funding, for example. So when people talk about primary liver cancer globally, people tend to talk about hepatocellular cancer, how it's the second or third commonest cause of death in humans from cancer. And how it is so important, and of course it is. But And then when people talk about primary liver cancer rates rising and HCC rising, but for example, a study that was done by HCC UK showed that between the late 1990s and late 2010s, primary liver cancer cases in the UK had tripled. And in that period, there were over 60,000 deaths from primary liver cancer. And even specialists in the field assumed that they were all predominantly HCC. But actually what they found was that 40% were intrahepatic cholangiocarcinoma. And so when you're trying to get funding in an increasingly financially stricken environment to try and do research, it's important that resources are put where there is a significant issue. And so it's important that cholangiocarcinoma is not deemed anymore to be an orphan disease, not numerically important. I mean, obviously it's important to every individual who suffers and their families, but globally as a disease burden, it is significant now. And that message needs to be got across, so appropriate resources are channeled towards it.
Fantastic. Thanks, Shahid. We actually have a question that's come through on the chat here. I'll just read out the question. If both CCA and CLATSKIN are mentioned in the documentation, while both have different codes, how do I differentiate? Or do I have to consult the physician for further detailing in each file? As it is most of the time mentioned both in a single admission, or do I have to code it as both as it describes different sites? I don't know if Andrew, you would have any insight to that.
No, I think Shahid probably would be better to answer that one. Sorry.
Yeah, I think so. So historically, before ICD-11, the issue was that if it was CLATSKIN, well, so this is a problem. So really, it shouldn't be called CLATSKIN. CLATSKIN is a term to describe perihilar cancers based on an American pathologist called Gerald CLATSKIN, who first sort of published a case series of these in the 1950s. And then the CLATSKIN name was attached to perihilar cancers without really strict anatomical definitions. And then the issue was that, so people were calling perihilar cholangios CLATSKINs, and there wasn't a code for perihilar or CLATSKIN, but if it was called CLATSKIN, it was incorrectly coded usually to intrahepatic. But with its, I don't know who was asking the question, whether it was someone who works in the registry or, but my answer would always be that if you're unsure, please do talk to a senior member of the tumor board or the MDT to clarify the anatomical location of what that should be coded as. And if it is and we should avoid the word CLATSKIN, and if it is a perihilar as anatomically defined, then with the new coding system, it should be defined as perihilar, and there will be a separate code for that. But if there's any doubt as to which of the subtypes it is, please do talk to a senior clinical member of the MDT team, the radiologist or the surgeon probably, or the hepatologist.
Okay. So leading on from that, what do you think the clinical community could do to encourage more accurate coding and recording of CCA at the diagnostic level?
Yeah, absolutely. So I was going to say, to follow on from that, what we have done is working through the Cancer Outcome Services dataset, which is the largest cancer dataset in England. We've added in a new data item which specifically defines the 3 subtypes of cholangiocarcinoma. So working with the experts within the MDT, you can not only use ICD coding, but you can also use a separate coding now to sub-define the subcategories as well, to make recording much more accurate. In addition to that, within the cancer registry world, what we've done is Professor Khan actually came out to a large conference we held 18 months ago to talk to the registrars to make sure that when we are getting data coming in, they understand how to record that, so that we've got national data that's far more accurate than what it has been in the past. So we're not relying on incorrect ICD coding. And also over the last 2.5 years, we've been working very closely with the molecular labs as well, so that we now get a feed coming through from all of the molecular labs. So if the cholangiocarcinomas have a molecular diagnosis, we will get that data coming in as a additional source of information, which we can then put against the patient's record, so that we can be as accurate as we possibly can for cancer registration in England.
Fantastic, thank you, Andrew. And I'd just like to open up to discuss with the panel if they've got any questions for each other here at this point.
I've got my hand up physically, not my little yellow hand, Helen. Yeah, I have actually, and it's just come up from something that has been mentioned today. And I'm not sure if it's just specific to the UK, maybe it's across Europe, but this is the use of the term primary liver cancer to mean only hepatocellular carcinoma, when actually there are 2 primary liver cancers. And I've just had discussions with the British Liver Trust and with various other groups, because it's been Liver Cancer Awareness Month. And yet again, cholangiocarcinoma is nowhere. And despite, you know, our best efforts from AMMF to push forward all the time, you know, cholangiocarcinoma is a primary liver cancer. There's this sort of, is not a synonym, is it? Primary liver cancer and cholangiocarcinoma do not mean the same thing, and that's it. And I just wonder, how can we educate people away from thinking primary liver cancer is HCC and vice versa?
Well, that's a good point. I mean, I try and do that. I was giving a talk on Monday at the Royal Society of Medicine on HCC, and many of my slides were actually about cholangiocarcinoma, just to emphasize the point that actually in cholangiocarcinoma is a huge chunk of primary liver cancers. You know, traditionally, Helen, you know, we were taught, and even now people are taught or told, or the textbooks say 80 to 90% of primary liver cancers are HCC, and 10 to 15% are intrahepatic cholangio, and then there's a few percent of very rare primary liver cancers like angiosarcomas or very rare things that one very hardly ever sees. But actually, it's more than 10-15%, and significantly more, as the UK data has shown. How do we get that message across? I don't know. John, do you have any hints and tips about how we do that?
Just lots of education, really. Actually, the big thing that we have over the last 10 years that we've never had before, really, is the MDT, where we can just week after week after week just rub it in, just repeat, just educate, and keep on going. And I think, from experience, it's not a bad way of passing across information, educating your colleagues about the new stuff that's coming along, and there's huge amounts of new stuff that's coming along, and this, you know, the business of diagnosis is absolutely critical. I thought you were going to tell us, Shahid, about the fact that in the past HCCs have been diagnosed just with.
Yeah. So I was just going to come and talk about that. Is that historically, that's exactly right. So historically, for a decade or so, we have been happy to treat HCCs without a biopsy. So it's about the only cancer, apart from the odd pancreatic cancer, I guess, where you're allowed to treat without histology based on imaging, and the imaging being that if there's contrast, which enhances in a triple phase contrast, and there's arterial enhancement and portal venous washout, you're allowed to call it an HCC. And that would be fine, except that many of those things that are then diagnosed as HCC purely based on imaging without histology are then ablated. So a needle is put in and burnt it. So you never get histology. Only a minority of these tumors are actually resected or transplanted. And it's when you do, if you do a resection or a transplant, you get the histology. But that's less than 10% probably of all HCCs in the Western world. And the majority of small tumors are ablated or treated with transarterial embolization, TACE or bland TACE. And again, if you ablate or you do embolization or intraarterial therapy, you don't get a biopsy. So you never know, you'll never know historically all those tumors that have been treated, what really were they HCCs? Were they mixed HCCs and cholangios, which is another subgroup, or were they cholangios? And there's data showing that in people who've been transplanted for presumed HCCs, the explanted liver actually had cholangios in it, for example. So there's a lot of data on that. But one of the encouraging things, and actually that this lack of biopsies is one of the reasons why our knowledge of the molecular pathogenesis of HCC may be a bit, and cholangiocarcinoma is maybe a bit behind other cancers, although we are making speed rapidly with that. But one of the positive things that has happened, and I'm pleased to see it with the trials that are being done now, is that biopsy is mandated in most trials of advanced cancer. And we are increasingly, our practice increasingly seems to be more and more to biopsy, because we have an understanding actually that we need to get histology. And then there's the whole issue of cancer of unknown primary as well, where people have liver lesions. We're not sure where the primary is. It's called CUP, and that again is probably a heterogeneous group of cancers, some of which at least are probably cholangio. But there is a trend to biopsy more, which is good.
We have a question in the messaging, which is, when would ICD-11 be implemented? And I know that can be different, obviously, depending on different regions. This individual's from Pakistan, and they're saying that many organisations are still stuck on ICD-9.
Yeah, so is Jacob on the call?
No, that might be one we could actually ask on Monday.
So Jacob, works directly in the institution, so probably best. But I did ask him this at a previous webinar, and it does vary from country to country, and different countries implement new ICD codings at different times, and that is another issue as to when we will have consistency internationally. Andrew, Andy, do you have anything you want to comment on about this, about why different countries implement at different times?
Yeah, I guess it's really down to the individual medical authorities as to when they want to upgrade to a newer version of a standard classification. I know that within England, for instance, NHS Digital are currently looking at ICD-11, but no decision has been made as to implementation. Once a decision has been made, I would guess it would take several years before all organisations within the NHS in England actually move over. But there has been no timescale addressed for that. And I know that he said that a lot of organisations are currently using version 9. It was only until about 2010, 2011 that most organisations in the US moved over from 9 to 10. So it can take a long time.
Countries that are using 9, do they have to go to 10, or can they go straight from 9 to 11?
I don't know is a simple answer.
And why does it take so long? Is it a very huge logistical issue in terms of IT and training and changing all your software?
Yeah, because they are so different. If you look at the difference between ICD-9 and ICD-10, they're very different. ICD-10 is easy to remember because they're just 3 or 4 digits, but the way the classification is done, you've only got a very small band of potential diagnoses you can have each one. So if you're looking at breast cancer, you've got C50, so you can only go C50.0 to C50.9, and then you're out of classification codes. Whereas with ICD-11, you've got many, many more, but the whole coding structure is completely different again. So it will take a while in order to bring that in, not just training within NHS trusts, but also with system suppliers making the difference so that those new codes can actually be recorded accurately.
Thank you. I'm just conscious that we're at time now, but I know, Jesus, you had your hand up earlier. Was there a point?
Yes, no, well, it's just a comment to the previous discussion. I fully agree that there is significant work to do in the education on the different subtypes, and also on the fact that cholangiocarcinoma is a primary liver cancer. But I want to also highlight that a significant effort has been done during the last years, and also that the specialists on the field are very sensitive on this. And for instance, for example, during the last years, there were a hepatocellular carcinoma summit, an annual meeting dedicated to hepatocellular carcinoma organized by the European Association for the Study of the Liver. And the title of this meeting changed 3 or 4 years ago to Liver Cancer Summit, trying to involve other kind of cancers, including cholangiocarcinoma. And cholangiocarcinoma has an important slot in this meeting. And I think this will help also to give visibility to this disease, similarly to other scientific societies. I will, if you allow me, I will add a very small comment. In terms of the biology of the tumors, we need to consider that cholangiocytes, which are the cells that form the biliary tree, are very heterogeneous along the biliary tree. So cholangiocytes within the liver are different to cholangiocytes from the extrahepatic bile ducts in terms of morphology, expression of markers, biology. So the tumors that arise in each anatomical location, by definition, might be different. This is important to emphasize this aspect. Also, these cells within the liver or outside the liver have a different microenvironment. So they are surrounded by other different cell types that can influence the biology of the tumor itself. And also we need to consider that the origin of cholangiocarcinoma tumors can be different. So what the definition of cholangiocarcinoma is the expression of markers of biliary differentiation only. But these tumors can originate from differentiated cholangiocytes, from hepatic progenitor cells, from progenitor cells in peribiliary glands, and also even from hepatocytes under transdifferentiation. The only thing that defines cholangiocarcinoma is the anatomical location and the expression of markers of biliary differentiation at the histological level. So all these aspects definitely impact on the tumor itself and on the features of the tumor, And well, this is something that I think is important to remark in addition to the previous very nice comments. Thank you.
But thank you very much for attending today. And again, very much thanks to our panel for facilitating a fantastic discussion.